Sir, mic is no.
So you are not audible.
Hello, am I audible?
Yes, sir. Yes.
Hello.
Yes, sir, audible.
Okay, man. Just a minute. Uh, Thank you all of you for giving me opportunity to present my work in this webinar. So, good afternoon, all the dignitaries and my dear audience. Myself, Dr. Himanshu Sharma. So, I have done my thesis on molecular markers and their characterization in different plants. So, today topic, am I audible, na?
Yes, sir.
Uh, Topic of today talk is the DNA marker technology and species conservation. So, firstly, Sabha has already explained. So, I will give the glimpse of the biodiversity. So, biodiversity is the life and diversity is the variety. So, it is the variety in the life. So, in simple term, it means the large variety of flora and fauna present around us, and they may be fungi, protozoa, or any of the life forms. So, the term biodiversity gained immediate acceptance as was brought to be attention in the 8th summit of the Rio de Janeiro. So, in this figure, what you think that on left side there is only one type of species and other type of the diverse species. So, biodiversity is the diversity of anything. Just like on the left side you are seeing a crop and other side just like forest which is full of biodiversity. So, so biodiversity is the diversity and it may be of different types. Genetic diversity. So genetic diversity as it means it is the different type of the collection of different genes and their diversity. So made up of the DNA and the building block of the organism and their interaction with the phenotypes that make it the trait. So genetic diversity can be measured at different levels including population, species level, and community level. So species level diversity may be of any species, or species may be defined as a group of individuals which can inbreed within a group. So you will find different species and their diversity is important to screen. So at that level, they may be species may be of different individuals or different plants or different communities. After that, last one is the ecosystem diversity. Ecosystem diversity is the diversity which is present at the different level of the ecosystem and interaction of different communities among the ecosystem. So ecosystem may be wildlife ecosystem, then it may be your aquatic ecosystem, or it may be any grassland ecosystem. So according to the species richness, there are different biodiversity hotspots. So richness is mainly depend upon the endemic species present in that area, and that may be more than 1500 plant species or any species. So now I come to my marker expertise. So that is mainly depend upon the genetic resources. So resources are the germplasm or any bank of anything. So these may be plant genetic resources, animal genetic resources, forest genetic resources, aquatic genetic resources, or genetic resources of microorganisms. So from the recent history, the cultivation is more than 10,000 years ago we have started, and by cultivating we are depleting the gene pool. So there is rapid need to characterize these gene pools and effectively use for the good gene pool for the plant breeders. So there are mainly four species: rice, maize, wheat, and potato, which made 100%, 70% of the food crops. So there is urgent need for these species to be characterized. So you already all know there are different type of the stresses: abiotic, biotic stresses. In abiotic stresses, temperature, water, salt, and radiations and chemicals. And in biotic stresses, it may be fungi, insects, herbivores, or other plants. So just like when there is a uh, different type of stresses, so plant behave in different way. So this is the just map. If we don't control our climate change, then up to this 2020, this is the scale of the climate change. So agriculture land is decreasing day by day. So when it will uh, 20280 due to climate change, temperature increase. So red areas are the conversion of the cultivation land to the barren lands. So if the temperature grows 2 degree 3 degree, then it will be cause the our populations or plant populations to restricted only. They don't they don't this they don't experience this the change in temperature, and they will the diminish their diversity. So they, and in a plant cell, this is the metabolic profiling. So there are millions of the pathways, and the scientists are uh, characterizing these pathways on the various approaches, just like phenome, metabolome, proteome, transcriptome, or genome. Phenome is your external diversity. Metabolome is the all the metabolic pathways or the products from the different gene expression. Proteome is the study of protein. Transcriptome is the study of expressed part of the genome, and the genome is your at the DNA level, different variations or different type of the SNPs you can find and use these for the trait development. So there are different type of databases which will store your this genomic information, just like DNA Data Bank of Japan, EMBL, and Genetic National Center for NCBI, National Center for the Biotechnology Information. So this, when you sequence any genome or any DNA, you have to submit these to the national repository so that anyone can use these information for their research. And this should be uh, free of cost. Anyone can use. If I will submit one DNA sequence, that can be used by any of the researchers around the world. These are the another different type of the databases, just like uh, SNP data or any type of the data. So, so genomics, it is the comprehensive set of the genes and their interaction. So this is we call the genomics. So genomics mainly is the sequence of DNA. So these days there is a lot of information generated from the sequence of the genomes, because there are Sabha has already told, I think, about the advancement of the genomes. Just like in 1960s, only Sanger sequence is available, and the one cost of doing one sequence is just like 1 lakh or 2 lakh. But these days it's very cheaper, and you can sequence your whole genome in just 10 of millions, 1 million. So there are different sequences, just like Roche, Illumina, Applied Biosystem, and these all the sequencer have their own limitations and uh, their advantages. Just like in Sanger, sequencing has been only DNA sequence method 30 years ago, but the NGS has the ability to process million of sequencing in parallel rather than 96 at that time. So these sequencers are they have saved our time and the funds also. So just like you can imagine this one in um, current time. You just in $10 you can sequence your genome, but by Sanger sequencing you will you have to put $2,400 for one run, which will cost about 2.5 lakhs to 3 lakhs rupees. So there are different genomic approaches for the improvement of traits, just like QTLs markers and their genes for the different stresses, and the tolerant genes or the different materials are used for the creation of gene pools in the plant breeding programs. So this is just a glimpse. Now I will give you some case study of my PhD thesis or my postdoctoral work. So this is the why we need the genomic markers. So advancement of NGS techniques have generated a lot of data, and that needs to be characterized. And in 1 week or 2 weeks, you will get lots of data, and you will go for the SSR markers or the intron markers or the SNP markers by comparing different genomes of the different species which you are targeting. So the molecular marker in layman term, molecular marker is the sequence which is present on the genome at specific location, which will use to um, compare the DNA of different species, different individuals. So marker जो है that is present on the DNA of is the your DNA, and it has unique sequencing which we will use to characterize the species, populations, or any type of the material you got. So a marker should have different properties, just like it should be highly polymorphic. Highly polymorphic means it should reveal the polymorphism. Codominant inheritance means it will reveals your heterozygosity, and it should be frequently present in the genome. It is easily means I am using it here in Shulini, then it will be easily accessible to you, and it should be highly reproducible and easy exchange, so that if I am doing, you can also do this type of the experiments in your lab. So there are different type of the, just like generation of your mobile signal markers are also in their advancement generation, just like RFLP is very previous type of markers. Then AFLP, RAPD, VNTRs, microsatellite SNPs. SNPs are the latest type of the markers. But the beauty of marker is that they have their own limitations and their own advantages. So their applications are the germplasm characterization, genetic characterization of transformants, organization of phylogenetic analysis, trait association also. So RFLP is the primitive type of marker in which we restrict our DNA with the enzyme, and then what, depending upon their deletion or insertion of any uh, sequence in the that part, that will give you the characterization of that species. So these are advantages, that these are codominant markers, and they have the uh, measure of the variation at DNA level, and disadvantage is that they need large amount of the DNA. Then AFLP is the modification your RFLP and RAPD. So the beauty of these marker is that if you don't know any DNA sequence or sequence information, then you will go for these primitive type of markers. And disadvantages of this marker is that it reveals only the dominance. It don't reveal the homozygosity. It reveals the homozygosity, but it don't reveals the heterozygosity. RAPD. So RAPD is also one type of primitive marker. They are fast, relative, and inexpensive. Everybody can use these markers in their labs if they have PCR or the GelDoc. So these markers also, their disadvantage is that these are markers are also dominant, and they reveals the homozygosity. Then after that there are SSR markers. These are white markers. So these are simple sequence repeat markers, and these sequence are repeated from 2 base pair to 6 base pairs. So these are highly variable, fast evolving, and codominant, but they are relatively expensive and time consuming. And after that, we have SNP markers. SNP markers are the variations in the single nucleotide polymorphism. So these markers are only we can do by the sequencing of genomes. And after sequencing, we have to compare these sequencing with the reference genome. Reference genome means If I am sequencing a project plant which has no information, then I will compare this with the already sequenced genome, just like Arabidopsis or eucalyptus, depending upon your plant which you are targeting. Then short tandem repeats, these are the 2 to 16 type repeat type of the markers, and they are mainly used in the forensic labs, fingerprinting of different individuals. So this is the comparison of different markers, how these are benefit or they benefit to us, and how they have the disadvantages. So in my PhD, I have targeted two type of the markers: intron based markers and the SSR based markers. So firstly, the approach is that we have done the transcriptome sequencing T clone, that is HV39, which is have high yield, and I also mined the public domain data from the NCBI. Then we have pooled all the data and then mined the SSR markers, and from the other side mined the intron targeted markers. So then we will use these markers for the amplification and validation in different T plants, and after that we have done the gene ontology. So gene ontology is the functional characterization of the marker containing sequences. So this is the T clone, which we have total RNA was isolated and then converted into the cDNA, and then sequencing was done on the Illumina. This is the in-house facility present at IHBT, and we have uh, done the transcriptome sequencing, and we will get the 12.4 million clean 11.2 million clean reads. So you can see 11.2 million clean reads which we will get from these sequencing. If this is the magic of this sequencing evolution. So we have used these sequencing for the mining of SSR markers, and we have well able to mine more than 6,000 markers were identified, and we correct then we will do the functional annotation of these markers. So functional annotation is that marker containing sequencings are blasted, or the sequence search against the known database which we will already have. So these will go for different categories, just like biological function, cellular function, and the molecular function. So from these we have pulled out some of the important information related to T and use these in, sequencing, just like they have help in the secondary metabolite genes. So we characterize these genes in the different T clones and find out these genes which are important and make some pathways which are related to the T. So same the other method is the geo enrichment. So geo enrichment method is that same, they are comparing the marker containing sequences with the this database, and this will give the highly enriched. The red one are the highly enriched, and how they are correlating with the different pathways. So this is the another method of the functional annotation. So this is the ILP containing uh, sequencing, and then finally the one database is the Kyoto Encyclopedia of Gene and Genomes. So this pathway also gives some important information of the important pathways, just like pathways of the secondary metabolite, metabolism, and other type of the genes. So we have done the same for the ILP markers, and then we have blasted against the transcription factor database. So transcription factor are the sequences present on the DNA where the transcription factors bind, and they will regulate or enhance the transcription or translation. So after that, this is the pure genetic fingerprinting work. So from this 6,000 markers we have characterized, we have synthesized approximate 280 markers, and from that we have got the 112 markers which are polymorphic, and we have characterized in the 12 samples 18 samples of the T. And also the beauty of these markers, they are the cross transfer. So these are the cross transfer individuals which has different species, just like I'm working on the Camellia sinensis. So these are the Camellia japonica, Camellia rosiflora, and Camellia sessenqua. So the transferability is more, means their genome sharing is more from these markers we can. So we can use these markers for the characterization of the wider gene pools of the tea. So markers application is that you can, if you have some information present in one form, you can use for the wider application of these markers in the other species or other communities also. So the again in on the same way we have used the your intron targeted markers, and we have synthesized 500, more than 500, and we got approximately 183 markers polymorphic. So these are the fingerprints which they are talking that markers should be polymorphic. So this is the polymorphic, means marker should be polymorphic. It has different bands present in the different individuals of the tea or any plant you are targeting. So after that, in thesis, so this was the study, and we publish it in the American Journal of Botany, and one is the plant breeding, and these all the international journals. So this one, intron targeted markers, was the first report from the world which we have used these markers. So these hypothesis is that we have a very low to moderate genetic diversity was existing in the tea clones present in the Kangda region of the tea. So this the average genetic diversity was 60 to 70%. Then after my postdoc work, where Sabha has told, I think told we are working on the amylose content. So there we have a population for the resistant starch. So different genes and alleles are responsible for controlling the starch present in wheat. So we have used different markers and sequencing techniques to the markers, and the mutation as certain in this study will be helpful for the uh, genetic improvement programs of the wheat. So this work was we have done in my lab where Sabha has done his PhD. So we have characterized 100 individuals of the wheat and mined different novel markers which we will use for the genetic diversity. So these three colors are representing that three clusters are present in the present gene pool. So this is in clear cut, you can see that this is the clear cut application of these markers. And these are the beauty of marker is that these are all the different allied species of the wheat, and this is the two clusters which are representing the Indian wheat varieties. So marker, so the good marker is that it will reveal or differentiate the species, and it will differentiate the clusters of the individuals present in the your germplasm. So one other, we have done other studies, just like we have done non-coding based RNA based markers in wheat. Then we have characterized some intron targeted markers for the starch biosynthesis in wheat. So these are all studies we have done in IHBT. And one last is that we have characterized the tilling sequencing. So tilling is that firstly we will mutate our population with some EMS mutant based, and then we will screen those mutations present inside the gene pool. So we will able to score 13 mutations which we have published last years in molecular biology reports. So we were able to target these alpha amylase and three of the transcription factors which are important, and we have find some of the mutations which and some of the students are carry forward these work for their PhD thesis. So this is very important information, and that can be targeted to find some of the information related to the regulation activity of these genes. After that, some of the work I have done on as ma'am already mentioned that I have got some Finland population. So he has some population in the ambrosia. So we have used chloroplast based markers. So chloroplast based markers are used for the phylogenetic analysis of the wild species. So similarly, we use these markers for the characterization of the onion species in India, and that we have published in Genetic Resource and Crop Evolution. So I am also serving review editor or associate editor in different journals, and then I have some projects. And so application in plant genomics is the last is gene identification and cloning and genome manipulations, then markers assisted selection, comparative genomics, QTL analysis, association mapping, and trait dissection. So conclusion is that the power of omics is the omics is central. And in the last slide what I have done, the proteomics, metabolomics, phenomics. So the power, this omics has the power to feel and fuel the world by the research. Okay, so this is I have all published 50 research papers in different journals. So thanks to Sabha to give me opportunity, and principal ma'am, Dr. Dr. Archana, I think, na, for inviting me for the invitation, and then students from the MDM college to listen me, and my funders, DBT and Department of Science Technology, and Dr. Joy K. Roy, where me and Sabha was the combined fellows at that time. So thank you, ma'am, and thank you all the dignitaries which I have left. Thank you all.
If anybody have questions, please put on chat box.